Good afternoon, everyone. I welcome all of you to this series of webinar of stem cell and regenerative medicine. So here we are again in front of you for our next informative second session on this series. And I hope all of you are healthy and safe at your home. Okay, and let's before we start the session, I would like to request you to take part in the poll so that we understand that how are you feeling today. So I'm going to start the poll. So please quickly register your responses so that we know that okay, you are completely geared up to have this session. Okay, and certainly you'll feel very happy and enthusiastic after attending this session because it's gonna improve your knowledge and like it will increase your knowledge for induced pluripotent cells, stem cells in all kind of aspects. So you'll certainly feel very happy, great after that, and I'm very happy that most of you are feeling great. So this poll is to make you familiar as well with the polling process, which is a integral part of our session, and that's how we understand. So we conduct the poll for the subject related topic, and then we understand that how much you are able to understand this topic. So please register your responses quickly. So 5 more seconds for poll to get over. Please register your responses quickly. I expect all the attendees here should take participation in poll. So please register your responses. So poll has been ended. So I'm very happy that most of you are either feeling great or they are feeling good, but all of you will feel great after this session. So with this introductory session, I would like to welcome our speaker of the day, Dr. Ankur Gautam. So a brief introduction about Dr. Ankur Gautam, as you are very well aware about him. And but Dr. Ankur Gautam did his bachelor's from MDS University, Ajmer, Rajasthan, and then later he did his master's and doctoral degree in biotechnology from Punjab University, Chandigarh. After finishing the master, he joined PhD in CSI Imtech, Chandigarh, and he joined there as a postdoctoral researcher, and there he worked for 9 long years as a research associate and later as a project scientist. So in January 2020, we welcomed Dr. Ankur in Merck High-End Skill Development Center and CSR Institute of Microbial Technology, Imtech, Chandigarh as a scientist. Currently, Dr. Ankur is working as a lab manager at Merck High-End Skill Development Center, Imtech, Chandigarh. So with this same thing and a lot of enthusiasm, I welcome Dr. Ankur for a wonderful session to present in front of you.
Yeah, thank you, Shubham, for this introduction. So, Shubham, please confirm if I am audible and my screen is visible to you.
Yes, Ankur, you are audible and the screen is clearly visible to all of us. Thank you.
Okay, thank you. So, if you think that there is some problem at my end, if my voice is breaking or if some problem in my slides, please inform me on my WhatsApp so that immediately I will rectify this.
Certainly. Yes, I'll do that.
Yeah, so thank you so much, all the participants, faculties, postdocs, PhDs. I welcome you all for today's webinar. So as you can see on my screen, so we have been receiving uh, feedback from participants that we should have a webinar series on stem cell and regenerative medicine. So students' feedback is very important to us, and we considered those requests, and we started this webinar series on a very interesting topic, stem cell and regenerative medicine. So we have already conducted one webinar on 21st of June, Introduction to Stem Cells, where Shubham explained very nicely on stem cells, their properties, their characteristics, their applications. And today I will take you to the journey of induced pluripotent stem cells. So I have prepared my slides in such a way that you will enjoy all the slides. I will take you all through the journey of induced pluripotent stem cells. So I will give you an overview how these cells are discovered, what are the different application of induced pluripotent stem cells, and what is the future of this induced pluripotent stem cells. So I will give you a snapshot of everything, and probably the last webinar in this series will be on 19th July, where all the major applications of stem cells in healthcare will be discussed by Ms. Aparna. And so as I told you that one webinar we already conducted, today is the second, and on 19th July we will have our final webinar in this webinar series. But this is not the end. We always have one webinar series after another. So we will have an in next webinar series with next set of topics, which we will give you information during the next webinar on 19th July, that what will be the next webinar series, and we will conduct these webinars after alternative week, right? After every alternative Monday, you will hear from us on a different topics. So as Aparna mentioned in chat box, we have a LinkedIn group. So this LinkedIn group is for all the faculties, academicians. So far we have around 4,700 members. I request all of you to join our LinkedIn group. I request Aparna one more time, you can share the link in the chat box. So those who haven't joined yet, please join our LinkedIn group. And we are updating all the information regarding our certificate courses, workshops, trainings on our LinkedIn page. And we will also separately send you email also, so that we will keep you updated regarding our upcoming events. Now coming to the today's webinar, it is on induced pluripotent stem cells and overview. So in the next 40-50 minutes, I will discuss all about induced pluripotent stem cells. But before I move ahead, I would request Shubham to ask poll question 1 and 2. So I think I request Shubham to ask question number 3 in the poll first, because I want to know how many you have a basic knowledge of stem cells, right? So Shubham,
Sure.
please ask question 3.
Okay. So participant poll is live for question number 3. So I request all of you to please participate in the poll. And the poll question goes as: identify cells in dish. Stem cell, cancer cell, somatic cell. So you can see the picture and then you have to identify.
The picture is on your screen.
Yes. Please register your responses quickly. I want the maximum participation over here. And it will help us to understand that where is your knowledge and how we have to take the session further, right? What would be the track which we will be following? So I request all of you to please register your responses so that we have maximum responses from all the participants. Okay, another 10 seconds. Okay, so I'm going to end the poll now. So Ankur, 77% of students have chosen the option A, stem cells.
Yeah, great. That's the correct answer. So thank you for responding. So now I have a basic idea that you know about stem cells, right? So I asked Shubham to ask question 1 now.
Okay. The poll is live now for question number 1. So I request all of you to please register your responses. Embryonic stem cells are totipotent, pluripotent, or multipotent. Please register your response.
Yeah, Shubham. So how many students responded to what option?
So Ankur, I'm going to end the poll. So uh, there is a mixed response. So 57% have chosen the option B, pluripotent, and doesn't have chosen the option A, totipotent.
Okay. So no worries. The correct option is B, pluripotent. So embryonic stem cells are pluripotent, and I will discuss in my subsequent slides, so don't worry. I just want to know how many of you have knowledge of pluripotent, totipotent, and multipotent. So that's why I asked these questions. No worries. So let me start the today's webinar. I will discuss gradually by introduction, discovery, characteristics, pros and cons, and then applications. So you all know about stem cells, right? Stem cells are special cells. They are, undifferentiated cells. They have capability of renewal themselves, and they can differentiate in any cell type, right? All our body cells can be developed from stem cells. So you must have heard about this, right? This is very basic, and this is very nicely covered in our previous webinar by Dr. Shubham, that stem cells are the cells which have renewable capability and they can differentiate into any cell type, right? Now, as you can see in the picture, so in our zygote, the cells, they are totipotent because they are capable of giving rise to any cell type, both embryonic and extra embryonic tissues. So only cells in your zygote, they are totipotent. But once the blastocyst is formed, inside the blastocyst there is an inner cell mass. And the inner cell mass is nothing, they are made up of embryonic stem cells. Now these embryonic stem cells, they are pluripotent because they are capable of giving rise to all cell types of the body, right? But they cannot develop extra embryonic tissues like placenta, right? So that's why they are known as pluripotent. So always remember, the zygote cells in zygote are totipotent, while cells in your inner cell mass in blastocyst, they are pluripotent, and those are embryonic stem cells. So I hope you understand the difference between totipotent and pluripotent. There is a marginal difference. Totipotent cells can give rise to all the cells, embryonic as well as extra embryonic tissues, right? All cells of body and extra embryonic, which contains placenta. But pluripotent cells, they can give rise to all cells of the body, not the extra embryonic tissue. And then if you go down, then multipotent cells. These cells are capable of giving rise to all cell types of particular tissue or organ. Like if a particular if heart stem cells can make heart cells, right? If you have cancer stem cells, they can give rise to cancer tumor cells, right, of a particular tissue. And then finally the highly differentiated cells, they cannot differentiate further. So if you go down from totipotent to pluripotent, multipotent, and nullipotent, their potential of potency, cell potency is gradually decreasing towards totipotent to multipotent, right? This is the basic difference. Now coming to the pluripotent stem cells, right? What are pluripotent stem cells? So as I discussed, only your embryonic stem cells, they are pluripotent because they have capability to give rise to all your body cells, right? Now what happened? This is your fertilized egg. Then from this fertilized egg, your zygote will be formed, and from zygote, blastocyst will be formed, and from blastocyst there is inner cell mass, and those inner cell mass contains your embryonic stem cells. Now this embryonic stem cell, since it's pluripotent, it is undifferentiated and pluripotent. What does it mean? It means that these embryonic stem cells can give rise to all your body cells, and all these cells are well differentiated. So undifferentiated pluripotent embryonic stem cells can give rise to all differentiated body cells like heart cells, your muscle cells, your fat cells, your blood cells, your lung cells. All organ of your body, they are developed from embryonic stem cells, right? So these embryonic stem cells, they are very, very useful. Or I can say the pluripotent stem cells, they have huge importance because they have capability to develop all your body organs. They are useful to treat many diseases, right? And they have huge potential in regenerative medicine because any damaged organ, any damaged cells can be treated or replaced by the pluripotent cells. We can develop a healthy cells from pluripotent and we can replace the damaged cells. So that's called is regenerative medicine. So regenerative medicine is nothing. So when you have a damaged cells or damaged organ, so you can replace the damaged cells or damaged organ with a new set of cells which can make the new organ. That is regenerative medicine. And pluripotent cells, they have potential to develop any body cell type which can develop the whole organ later on, right? So that's why there is a huge potential are there in pluripotent stem cells, and they have a huge application in regenerative medicine. Now coming to the embryonic stem cell research. So you know that embryonic stem cells, they have capability to develop the whole embryo, whole body, right? So once embryonic stem cells can develop the whole organ, the whole body, so it can be used for development of different embryos. And that's why that is a matter of big concern, because here we are using embryonic stem cells from the embryos. So we are actually sacrificing the embryos, right? And that's why there are lot of ethical issues are there in using embryonic stem cells, human embryonic stem cells. And this is banned, right? So we cannot use embryonic stem cells as such for our research because of various ethical issues, right? So once we cannot use embryonic stem cells, so how we can go in regenerative medicine era, right? How regenerative medicine will be, turned into reality without embryonic stem cell research? That is a question. So there was a major technological breakthrough in science happened almost 1.5 decades back. It was exactly the 2006, year 2006, right? Now what happened when scientists gave an idea that you can turn any cell of your body into a stem cell. Right, let me repeat it. You can convert any cell of your body into a stem cell. Wow, is it interesting? Any body cell, your skin cells, your heart cells, your muscle cells, your nerve cells, you can convert them into a stem cells. It's really interesting. So this is your body cells, somatic cells. You can convert them into embryonic stem cell like cells. So this is really interesting, and this is really a major technological breakthrough in science, right? This breakthrough actually changed our thinking about how cells work. Nobody even thought about that our normal body cells can be converted into cells which are very much similar to embryonic stem cells, right? But just imagine, because of this, you need not to work on embryonic stem cells, because you can develop your own embryonic stem cells from your own normal body cells. So there is no need to use embryonic stem cells, no need to sacrifice embryos. So you can imagine how big breakthrough it was, right? It's really changed the science. It's a revolutionary breakthrough, I can say. Now, this great person in the picture, Shinya Yamanaka, who actually is a MD and a PhD by profession, and he is working in Kyoto University in Japan. So he was the person who gave first time this idea that you can convert your normal body cells into embryonic stem cells or similar to embryonic stem cells. And this was the landmark discovery in stem cell research, right? So I will give you an overview how this discovery was done, how Shinya Yamanaka performed their experiments. I will tell you everything gradually, and I hope you will enjoy this. So those who already know about this, you can just recall, and those who didn't know about anything about this, you will enjoy this, okay? So first time the concept of induced pluripotent stem cells was given by Shinya Yamanaka in 2006. Now, as you can see, what happened in normal process? From fertilized egg, you got zygote. From zygote, you will get blastocyst. From blastocyst, you will get embryonic stem cells. And I told you, embryonic stem cells, they are undifferentiated, right? And since they are undifferentiated and they are pluripotent in nature, they can give rise to any cell type, right? Any cell type. So now you can say the undifferentiated cells can be converted into differentiated cells. This is known by decades. Everyone knows about it. What's new in this? The novel thing is that can differentiated cells be converted back into undifferentiated cells? So this was not known before, right? This was not known before. No one thought about this. That is it possible that differentiated cells can be converted into undifferentiated pluripotent cells, right? So this was the person, or this is the person, I can say, Shinya Yamanaka, who first time told that yes, this is possible. Even the differentiated cells can be converted into undifferentiated pluripotent cells. And these cells are known as induced pluripotent stem cells, iPSCs, right? These cells grow like a colony, as you can see in the picture. This colony is roughly consists of more than 500 induced pluripotent cells, and the size of one pluripotent or induced pluripotent cells is around 10 micrometer. So roughly 500 cells are there in this colony, and these cells are grow like different small colonies like this, right? So what was the hypothesis? behind this great discovery. Yamanaka hypothesized that differentiated somatic cells. Please, I need your attention over here. Differentiated somatic cells, they can be reprogrammed. They can be reprogrammed into pluripotent cells with different set of genes which are important for the embryonic stem cell identity. I'll repeat the hypothesis. Differentiated somatic cells of our body, they can be reprogrammed into pluripotent cells which are similar to embryonic stem cells with a certain number of genes which are important for the embryonic stem cell identity. Right? So Yamanaka gave this hypothesis, and he thought that if this hypothesis proved right, then there will be two big advantages. First, there should be no embryo requirement. We can bypass the ethical issues. And we can create patient-specific pluripotent stem cells, right? Suppose if person is there for cardiovascular disease, you can take the normal body cells of cardiovascular patient and convert them into pluripotent stem cells, right? And then these cells can be converted into again heart cells. So you will have patient-specific pluripotent stem cells to study, right? So this was the hypothesis, and then Yamanaka worked on his hypothesis. He provided experimental evidences that this can be possible. So how he did it? He actually identified different genes which are required for embryonic stem cell identity. When I say embryonic stem cell identity, what does it mean? It means that genes which are responsible for the self-renewal of embryonic stem cells, genes which are responsible for the pluripotency capability of the stem cells. So he identified almost 24 different genes which are required for embryonic stem cells to maintain its identity, to maintain its self-renewal, to maintain its pluripotency. So what Yamanaka did is, one by one, he transfer all the genes or deliver all the genes into normal somatic cells, that is fibroblasts, skin cells. So he deliver one gene in one time by retroviral delivery into normal somatic fibroblast, and then he evaluated whether the somatic cells converted into induced pluripotent cells or not. So this he did one by one with all the 24 genes and with different combination and permutation, right? And finally, after this experiment, he narrowed down from 24 genes to 4 important genes: OCT4, SOX2, KLF4, and CMYC. And he proved that just by delivering these 4 different genes into normal cells, your normal somatic cells will converted into pluripotent cells. So this he proved, right? And those factors, these 4 factors, these genes actually code for transcription factors, right? These are 4 transcription factors, and those are known as Yamanaka factors after this discovery, right? So first time in 2006, Yamanaka discovered that mouse normal skin cells can be converted into induced pluripotent cells by just delivering these 4 factors. So what he did, he just took skin of mouse and grow the skin fibroblast cells, these are somatic cells, then deliver these 4 factors into these cells, and this process is known as reprogramming. So now onwards, whenever I use the term reprogramming, it means that delivery of these 4 factors into the cells. So when Yamanaka reprogrammed normal mouse skin fibroblast with these 4 factors, after 2 to 3 weeks, he observed that the small colonies of induced pluripotent stem cells are started growing in the dish, right? Your skin fibroblast converted into induced pluripotent stem cells after reprogramming. And when he studied the characteristics of these induced pluripotent stem cells, he found that these cells are behaving very much similar to embryonic stem cells. So he took mouse, he took embryonic stem cells, he took fibroblast. So he grew embryonic stem cells and he converted this fibroblast by reprogramming, and then look at the morphology of embryonic stem cells and induced pluripotent stem cells. And he also studied the various other parameters of both cells, and he found that there is a high level of similarity in the morphology, in the growth properties, and in the expression of various biomarkers, which proved that the induced pluripotent stem cells, they are similar to the embryonic stem cells only, right? And then at that time, when Yamanaka proved and told about this discovery to different scientists, so scientists were surprised, right? It was very hard to digest for them that how could this happen? How the differentiated cells can be converted into pluripotent cells. So it will be very difficult for scientists to digest this, right? And now there is a debate started that it cannot be possible. He is crazy, right? But this happened with all the major discoveries. Nobody will, realize this at the moment. It will take some time. You have to prove them. So it was difficult for the scientists to realize this, that okay, this can be possible, because nobody knows about it before, right? But in 2006 only, Yamanaka published his findings in a prestigious cell journal, right? This was the paper. If you get some time, just go through this paper. And then after that, Yamanaka worked on human cells and he performed the same experiments. He took biopsy of the human and cultured the human skin fibroblast, reprogrammed them, and then he observed that after 3 to 4 weeks, again small colonies of induced pluripotent stem cells start growing in the dish. So he proved that by the similar programming, by the similar delivery of 4 factors into human fibroblast, induced pluripotent stem cells can be generated, right? And again, he published this finding also in Cell Journal, back-to-back 2 papers in 2006 on mouse fibroblast and in 2007 on human fibroblast. Then scientists started realizing this, and they also started working on those experiments, and soon this was observed that in the same year two other groups they have also performed the similar experiments with similar or slightly different factors. They also found that yes, the somatic cells, the normal cells, can be reprogrammed and induced pluripotent cells can be generated from normal cells also. Now scientists started reproducing Yamanaka's findings, right? So this is how the great discovery was carried out, and because of this landmark discovery, the Science Journal declared this reprogramming as Science Magazine 2008 Breakthrough of the Year, right? Isn't it very interesting? And it's really a major breakthrough, right? And because of this fantastic work of Yamanaka, in 2012, he was awarded with a Nobel Prize, right? Which is the biggest and prestigious award in science. So these are the few images of Yamanaka receiving the Nobel Prize in Physiology and Medicine in 2012. So this is how iPSC concept came into existence. So let me give you a very simple example and tell you what is the basic concept behind induced pluripotent stem cells, right? So you can take any somatic cells from your body, right? You can take your muscle cells, you can take your fat cells, you can take your blood cells, you can take your fibroblast and just induce them. Just deliver these four factors and they will convert into undifferentiated, and pluripotency will be triggered in these undifferentiated cells, and those cells are known as induced pluripotent stem cells because they have characteristics of embryonic stem cells. Now if you provide right environment, they can differentiate into all your body cells like embryonic stem cells. So by just delivering of four factor into any normal cells, you can induce pluripotent stem cells which are very much similar to embryonic stem cells and can differentiate into any body cell type, right? So I can give you one example. This is very much similar. Suppose you have a cake, but you don't want to eat cake, rather you want to eat cookies. So you, if you can convert this cake back into flour and eggs, and then you can make again cookies out of it, right? So this is exactly same happen in iPSCs. Already differentiated somatic cells can be converted back into undifferentiated pluripotent cells, and those undifferentiated pluripotent cells can give rise to all other body cell types, right? So this is the basic concept of induced pluripotent stem cells. I hope you understand the concept now. Now question is, what is so special about iPSCs, right? So iPSCs are like embryonic stem cells. They have capability of cell renewal. They have capability of clonality, and they can give rise to any body cell types, right? Cell renewal means undergo numerous cycles, keep on dividing. Clonality means they can divide and generate similar cells. And differentiation means when you provide the right growth factor, right environment, they can differentiate into any body cell types. Now there is one more interesting thing about iPSCs. Do you have any idea? All the characters PSC, they are uppercase. Why I in lowercase in iPSCs? Any guesses? So at that time, iPod was very popular. So Yamanaka thought that he should give name is iPSC where I should be in lowercase because of popularity of iPod. Isn't it interesting? Right? So that's why the I is in lowercase in iPSC. Okay. Okay, now coming to that, I will give you a brief history of iPSC, though I already covered it. So in 2006, Yamanaka first time generated iPSCs from mouse fibroblast. Next year, 2007, he did the same experiment in human fibroblast. In 2008, the alternate programming methods came into existence. In 2008 also, the disease modeling and drug discovery using iPSCs started, right? In 2012, Yamanaka and other scientists were done, they awarded Nobel Prize. And in 2014, the first clinical trial using human iPSC derived cells was started, right? So this is a short, brief history of iPSC, which I already covered. So now this time the question 2, over to Shubham. You can ask question 2 at this stage.
Okay, Ankur. So poll is live now for question number 2. So question number 2 poll is live now. What are Yamanaka factors? A. Transcription factors used for reprogramming. B. Physical factors affecting the growth of cells. And C. The growth factors. So I request all of you to please register your responses quickly. We have 20 more seconds left with us. Okay, so I'm going to end the poll now. So Ankur, I'm happy to tell you that 95% have chosen the option A, transcription factor used for reprogramming.
Yeah, that's the great. I think I hope that all of you understand the concept. So that's why I framed this question. Thank you, Shubham, for conducting this poll. Now coming to the next. Now you must be curious to know how these iPSC generated in the laboratory, right? Let me give you an overview. So first you have to take a biopsy samples or any normal cells, for example, skin biopsy. From skin biopsy, you can grow the fibroblast as a primary culture, and then you have to just deliver the 4 reprogramming factors by retroviral delivery or any other way you can deliver them, and then you have to wait. You have to grow those cells. You have to maintain the cells in embryonic stem cells media, and you will see that after 3-4 weeks, small colonies are started, they're growing in the petri dish, and these are partially programmed cells, and eventually later on they will mature into induced pluripotent stem cells. The whole experiment will take around 1 month or so. Generally, these after transfection of these reprogramming factors, in 4 to 5 weeks, you will see that colonies of induced pluripotent stem cells are growing in your petri dish. This is very simple. There is no rocket science in this. Now, question is, nowadays these iPS cell lines are already available with different commercial sources, right? So, how to maintain and how to generate iPS cell line? So, you have to take skin biopsies from any donor, grow the fibroblast in your petri dish as primary culture, then reprogram them, and then you will notice that there are certain changes in the morphology of the cells. Some cells are started growing in colonies, right? Then you can always stain live with TRA60 or SECA4 dyes, and these are the dyes which specifically stain the induced pluripotent stem cells. And then you can always pick those colonies and expand them. You have to expand specifically the positive stained colonies and expand them, and then you can actually passage them and you can grow them further, and you can look at the morphology of these cells under microscope. So you will see those cells which have very good morphology, like the rim of the colony should be very clear, right? And if you have a very good morphology of the colony, you can always write on the track. So how these iPS cell lines are obtained and they can maintain further, right? So you should be very careful that when you are maintaining these iPS cell line, there should be no cross contamination with the differentiated fibroblast. These all should be undifferentiated pluripotent cells, right? This is the main challenge with generating iPS cell line. Okay, next is, let me give you an iPS culture overview. There are three steps, like you have to grow them, you have to expand them, and you have to differentiate. So when you grow, you have to take biopsies, grow the fibroblast, and then reprogram them and generate iPS cells, right? This is the first step, growing and converting iPS cells. Then you have to expand them. Now, once you have purified iPS cells, you just pick the colony and expand them in large cultures. And then finally, when you have a sufficient amount of iPS cells, this is the time that you have to provide the right environment to differentiate them into your cell of interest. So by using these iPS cells, you can provide them a right differentiation media, right environment, so that they can differentiate whatever you want, neuronal cells, muscle cells, fat cells, bone cells. So you have to provide the appropriate growth factors and the environment so that they can give rise to that particular cells. So these are the three major areas or steps: grow, expand, and differentiation. Right? Now coming to the quality check. So whenever you are maintaining iPS cell line, you have to check the quality of these cells. What does it mean? Quality checks means you have to ensure the purity and quality of generated cells and their pluripotency potential. You have to ensure that they have pluripotency potential. You have to ensure that they are pure. There should be no contamination with the differentiated fibroblast, and their quality should be very good. And this can be done by looking at their morphology. Their morphology should be very much similar to embryonic stem cells morphology. And there are certain biomarkers which are expressed on embryonic stem cells. Those all biomarkers should also be present on induced pluripotent stem cells because they are responsible for the pluripotency. So you can go for immunohistochemistry and look at those biomarkers with whether they are expressing or not, right? And then you can see the growth pattern. It should be similar to embryonic stem cells. You can see the differentiation potential also. So once you ensure that they are pure, they have all the markers which are responsible for the pluripotency, their growth is very much similar to embryonic stem cells, like their doubling time and all these things. So these are all quality check are required at every step for looking at whether your IP cell line is good or not, right? Now coming to the application part, because now you know that these induced pluripotent stem cells can give rise to any body cell type, so they have huge potential in regenerative medicine. So they can be used disease modeling, they can be used for drug screening, they can be used for cell therapy. I will discuss one by one, right? First, let me give an overview of how iPSCs can be used as a human disease modeling. So suppose I can give you an example. If you are working on a particular disease and you want to understand the disease in a better way, what you do? Usually you work on animal models. You develop a disease into animals and see how animals are behaving, right? So animal models can be used to understand a disease in a better way, right? But because we have disease in human and we are developing in animals, though the animals like mouse or rat, they are very closer to humans, but still there are certain differences, right? So how about if you have a human cell model instead of animal model, right? That will be more physiological relevant, right? So how you can develop this human disease model? So what you can do, you can take a healthy individual and a patient. You take a biopsy sample from both these and grow the fibroblast, healthy fibroblast and disease fibroblast, and then convert them into iPSCs. So you will get healthy iPSCs and patient-specific iPSCs, or I can say disease iPSCs, right? Now you can grow them and expand them in a 2D culture. So in 2D culture, you know, the traditional cell culture is known as 2D cell culture, which we used to do. You can differentiate those iPSCs into, suppose you are working on neurodegeneration. So you can differentiate your iPSCs into neurons, right? So you will get healthy neurons and diseased neurons. Now you can compare their molecular signatures, their properties, their characteristics. This will give you an idea that how diseased neurons, they are responding or behaving differently than your normal neurons. And these are human neurons. And one step forward, you can further grow these 2D cell culture into 3D cell culture, and you will get organoids. So organoids are a type of 3D cell culture. So in organoid, cells grow in a different manner in 3D environment, and they will give you a feeling like of a small mini organ, as you can see in the picture. This pink color picture is cerebral organoid, right? So when you grow the neurons or neurological cells in 3D environment, they will give you a morphology of mini brain. They will grow like that only, right? So these control organoids and patient organoids, you can always have properties. They can compare their properties, and these 3D cell culture or 3D organoids, control and patient, they are much more physiological relevant model to study the disease rather than developing disease in animal models, right? You can always compare the properties of these 3D cell culture or 2D cell culture of healthy and patient neurons and organoids to have a better understanding of the disease. So that's why these iPSCs, they have a huge application as human disease model systems because they are human cells, they are derived from human cells. So nothing better than a human cells rather than doing it on animals, right? Once you have a human disease model, the next step is now if you want to develop a drug particular against suppose Parkinson disease, right? So what you can do, you can develop a iPSC model of Parkinson. You can always take biopsy sample, grow fibroblast, reprogram them, and these are your disease specific iPSCs. Now you can convert them into neurons, and now you have your human iPSC derived neurons where you can screen your drugs, where you can perform your toxicity scanning, and finally you can develop drugs. These drugs are much more efficacious because you directly tested and screen on human iPSC derived neurons, which are much more closer to the physiological relevance as compared to animal model systems, right? So this is how drug screening and toxicity screening can be done on iPSC model systems. Now next is the cell therapy. This is the most important. Now what you can do is, suppose there is a patient, you can take a biopsy and grow a skin fibroblast, reprogram them, generate patient specific iPSCs. Now these patient specific iPSCs, suppose there is a mutation in a particular gene and because of that there is a disease in the person. So in these patient specific iPSCs, those mutations can be corrected by genome editing. And once you correct those mutations, now you have a genetically corrected iPSCs, right? These genetically corrected iPSCs, they can be differentiated into body cell types, and again they can check for the quality, and then they can transplant it back into the patient. Suppose a person is suffering from you can say neurological disorders. So you can take the biopsy, grow fibroblast, reprogram them, convert patient-specific iPSCs by genome editing, corrected the errors, and these corrected iPSCs now again differentiated into neurons. Now these neurons, they don't have those mutations because you already corrected with the genome editing. So these healthy neurons, because they are derived from the patient somatic cell itself, so they can, after checking the quality, they can transplant it back into the patient, right? So here we are taking cells from the patient, and by converting into healthy cells, we are transplanting back into the same patient. So this is known as auto transplant. This is the beauty of cell therapy by iPSCs, right? There is one more scenario. So suppose you have a donor, you have a healthy donor, healthy individual, you can take fibroblasts from healthy individual, reprogram them, generate healthy iPSCs, and then check the quality, then differentiate into any cell types, cardiovascular disease for heart muscle, heart cells, for bone, cartilage cells, for diabetes, beta cells, and then you can again check the quality and then transplant back into the patient. So here you are taking cells from other healthy person and injecting into a different patient. So this is known as allotransplantation, right? So everything can be done. This is how cell therapy works in case of iPSCs. The only problem with cell therapy is that if you look at the whole process, starting from taking sample from the donor and to grow as a fibroblast, induce them as a iPSC, then quality check, then differentiation, then again quality check, and then transplantation. So from sample collection to transplantation, the duration of the time is allowed around 1 year. That is too much. Sometimes patient is so severely affected that they will die in this time. They won't survive. So time is the issue, and normally it is expensive approach. In general, for per patient, $1 million is the cost for the treatment, right? So these are two factors. Now, what scientists thought about that, how we can shorter the time period. So they thought that why not we can storage the iPSCs. We can generate iPSC and store them. So there is a healthy donor, take the biopsy or blood samples, convert them into iPSCs. These are the clinical grade iPSCs. You have to check the quality. And then after quality check, you can always store them and then distribute. So by doing so, you can save the time, right? Whenever a patient required the iPSCs, you can immediately distribute. Now the only disadvantage is that immune rejection, because you are taking cells from a different donor, right? And you are injecting into a patient. So you have to match the HLA haplotype, which is the tedious task. But scientists now have solution to this problem also. What is the solution? So they are identifying super donors which are homozygous, and I can tell you, I'm not going in detail, these donors when gave samples with, so these samples are compatible with most of the population, right? So there is minimum immune rejection if your donor is super donor and he is homozygous. We can discuss what is homozygous or what is the criteria of super donor some other time. So these super donors are very rare. Like if you scan a population of 1,000 or 2,000 people, you will get one super donor, right? But if you take sample from such kind of super donor, so that can be distribute to all the patients, and there will be a minimized immune rejections. So by preparing stocks, you can save a lot of time, right? And then you can directly differentiate the cell as per the patient demand, and you can transplant. So you can shorten the time and the expense will also be less, right? So this is the latest advancement which is going on in iPSCs. So now question is, iPSCs, as I told you, that they are very tiny cells, right? They are very tiny cells, but they have a huge potential. I told you their diameter is 10 micron, right? So they can convert into any cell type. They have a huge potential. So suppose now for till date, iPSC cells can be converted into different cell types: neurons, heart muscles, retinal cells, neural stem cells, cartilages, platelets, immune cells, and they can be used for different disease like neurological disorders, heart disease, eye disease, spinal cord injury, arthritic disorders, blood transfusion, and cancers. So there is a great future of iPSCs, but there are still challenges also. So now the next is, because the viral delivery, because of this viral delivery, these factor, the gene responsible for this factor, they are integrated into the genome. And because sometimes the procedure is so lengthy that during the course of the procedure, some mutation can be accumulated. So now scientists are looking for alternate ways of reprogramming. So they are using viruses which are not going to integrate the gene into the genome, right? Like Sendai viruses, or they are using plasmid also, right? And nowadays different approaches like direct transfection can be used. So instead of delivering the gene, now scientists are delivering the mRNA only. Scientists are delivering the direct protein, all the proteins, and scientists are also looking for small chemical compounds which can reprogram the cells. So these are the latest advancement, that instead of delivering through retroviral, can we deliver without integrating the genome? Can the same kind of reprogramming can be done by chemical compounds? Can specific miRNA or mRNA can be used, right? So these are the latest advancements which are going on as the alternative ways of reprogramming. Now look at the advantages of iPSCs. The biggest advantage is that you need not to require embryos. There is no ethical issues. iPSCs can differentiate into many cell types. They are suitable for auto transplantation. So they can be patient specific or individual specific. There is no rejection, no immunological response. They can be useful for studying mechanism of diseases. They can be used for drug screening and toxicity testing. They are easily accessible and they are vastly renewable. So these are the advantages of iPSCs which make them so popular. Now there are certain challenges also. What are those challenges? The first challenge is that the efficiency of reprogramming is very low, 0.1%. But nowadays, due to the availability of different advances, scientists are working to increase the efficiency of reprogramming. The second challenge is viral vector for induction. Now, as I told you that scientists are looking for alternate ways for reprogramming. And the biggest disadvantage is that there is always a risk of tumor formation. Why? Because in 4 factors, if you look at the 4 factors, the 2 factors, KLF4 and c-MYC, they are proto-oncogenes. They are oncogenes. So once you increase the expression of these genes, they will induce tumor formation. This is the challenge. Now scientists are thinking, can we reprogram the cells without delivering c-MYC and KLF4? So without using c-MYC and KLF4, scientists are trying to find out ways to reprogram the cells so that there should be no risk of cancer development. Next is economic burden. As I told you that this treatment is very, very costly, but scientists are looking for different ways to cut down the cost. And then time. So as I told you that it's time consuming, but by making pre-prepared iPSC stock, you can decrease the time significantly, right? So these are the pros and cons on iPSCs. I hope you have understand this. Now, there is other challenge is rejection. So rejection can also be avoided by shutting down or by silencing the expression of MSC class 1 and class 2 molecules. Now scientists are working if we can decrease the expression by genome editing of MSC class 1 and class 2 so that there should be no problem with the immune rejection. So still iPSCs are in preliminary stage. It has to be a long way to go, but definitely there is no doubt that the future is very bright and they have a huge potential. If we work on these challenges, then it will very, very good for science that we can have answers or we can have solutions of very or many genetic disorders or different diseases can be treated with iPSCs if we overcome these challenges. Now you must be thinking that what is the current status of iPSCs in clinic, right? So let me tell you, there are around 54 clinical trials are going on for different diseases in different countries. The first clinical trial on iPSC was started in 2014 in Japan by Rankin Institute. They have used iPSC-derived retinal pigmented epithelium, and this trial was against age-related macular degeneration, which is a common problem in old age blindness, right? So these 54 trials, they are going against different diseases, right? The most common diseases are neurological disorders, followed by cardiovascular diseases, and then ophthalmologic diseases. If you look at the countries, the US is the number 1 Maximum clinical trials are going on US, followed by France and Germany, and the rest are the other states. So this is the overview of clinical trials on iPSCs. Now there is a very good video which I want to play for all of you. So let me play that video for you. So Shubham, please confirm if you can see the video on the screen.
Ankur, yes, Ankur, it's audible clearly.
In 1996, the world's first cloned mammal, Dolly the sheep. In 1996, the world's first cloned mammal, Dolly the sheep, was born. Her birth proved that the mammalian genome could travel back in time. Placed into an egg, the DNA of a specialised or differentiated cell gained the powers of an undifferentiated stem cell. Inactive parts of the genome were reawakened and directed the development of a whole new animal. Dolly's birth launched a race not to clone people, but to find a way to make human embryonic stem cells from a specific individual. Embryonic stem cells can make any sort of body tissue, so this promised to provide tailor-made human cells for research and transplantation. But making cloned embryonic stem cells in the lab proved very difficult, and on top of that, in 2005, the field was bruised by a high-profile fraud. Around this time, Shinya Yamanaka finished his own experiments. He'd found a way to make cells similar to cloned embryonic stem cells, but without using eggs or embryos, making the process much more straightforward. His technique was so revolutionary, he worried other scientists wouldn't believe him. Using just 4 genes, Yamanaka had changed mature mouse cells into pluripotent stem cells, cells that could then be used to make almost any cell in the body. Yamanaka named his reprogrammed cells Induced Pluripotent Stem Cells, or iPS cells. At first, the scientific community was sceptical, but once Yamanaka revealed the identity of the 4 genes, other scientists quickly reproduced his work. One of these scientists was Konrad Hochadlinger. He engineered his mouse cells to make green fluorescent protein whenever a crucial stem cell gene was active. Then he tried Yamanaka's reprogramming technique for himself. To his astonishment, his cells glowed green. They had indeed turned into stem cells. Hochadlinger and other scientists showed that these stem cells were capable of turning into all kinds of tissue in mice, including heart tissue, skin, and even sperm. Then came human cells. Just a few months later, more scientists showed that our own cells could also be rewound to a stem cell state. So much for cells in dishes. The ultimate test was to make healthy, fertile mice entirely out of iPS cells. Recently, iPS cells rose to this challenge too. Nature Methods has named iPS cells Method of the Year 2009 because of their promise as tools to study biology. Although iPS cells were first made some years ago, it's only now that they are beginning to be used in this way. Studying the cellular machinery that enables an iPS cell to turn into another cell type could help us understand what makes a nerve cell acquire the ability to transmit impulses, or a heart cell to beat. Researchers are also excited about using iPS cells to study human diseases in human tissue, likely to be a more accurate way to model disease than using mouse models. One of the first studies to use iPS cells to study disease was by Clive Svensson. He made iPS cells from cells taken from a young boy with spinal muscular atrophy, and then turned the iPS cells into neurons. This enabled him to study the differences between diseased and healthy brain cells. There are still a few problems with this approach. Not all cells that look like iPS cells have actually been thoroughly reprogrammed, so researchers are working on better ways to make and assess iPS cells and their offspring. And this work won't spell the end of embryonic stem cell research. Embryonic stem cells are still the best understood pluripotent cells, and it remains to be seen if iPS cells are entirely equivalent to them. But the iPS revolution has most definitely begun. In the last year, labs all over the world have published high-profile papers, and many research groups are starting to make disease-specific iPS cell lines. In the next few years, these new additions to the biologist's toolkit should start to show their full potential, increasing our understanding.
So thank you so much for watching this video, and this is all from my side. I hope you enjoyed this today's sessions, and probably I can take few questions right now. So there are many questions, which means that you all are very curious to know more about these technologies, right? So I can take few questions, but we will answer all your questions. We will send a question answer document after these sessions along with the PPT handout, where all the unanswered questions will be there, and we will address all your concerns. So let me take few questions. I can see few questions. Let me take randomly few questions. The first question is, CMYC and KLF4 are proto-oncogenes. How this is determined by scientists? Suggest a strategy for this. So, CMYC and KLF4, they are known as proto-oncogenes from decades, right? But at the same time, these CMYC and KLF4, they are required. They have high expression in embryonic stem cells because embryonic stem cells have to keep on growing, right? So that's why when this Dr. Yamanaka studied the different factors, he found that these two factors are also playing important role in pluripotency. So they incorporated these two factors in their panel. So that's why they incorporated. But the question is how this is determined by the scientist. So there are many molecular biology techniques are available, like you can knock out a particular gene and see whether your cells are proliferating or not, right? So to understand a function of any gene, you just simply knock out it and see the behavior of the cells. And this is the way to understand whether a gene is a proto-oncogene, whether a gene is a tumor suppressor gene, or whatever the function of the gene you can determine by just knocking down or knocking out of the cells. Okay. So next question is, let me see. Can you explain the HLA typing advantages and disadvantages of this lecture? So HLA typing is that whenever you go for a transplantation, whether a organ transplantation or cell transplantation, so if you are HLA typing, if you don't do HLA typing, then there is a high chance of rejection. Your body will reject the cells or the organ, right? So you must go for HLA typing. So we will have another webinar series on HLA typing because it's a very broad topic. So only I can say that whenever you are going for a transplantation, like your fingers, right? Fingerprints, they are not identical. So all the students sitting today attending this webinar, they are not identical. Similarly, the HLAs, they are 10,000 different HLA types are there. So you have to match HLA types, otherwise your body will reject it, right? So either you have similar HLA type or you have a super donor kind of thing whose cells or organs can be acceptable to your body. So HLA typing is very, very must for the transplantation. Now next is could we have used this approach in drug screening in clinical trials of vaccine too? Yes. So not in clinical trials. So most of the clinical trials, they are either based on cell therapy or regenerative medicines. But yes, for developing drugs, not in clinical trials, but in preclinical studies, these iPSC based models are being used for different purposes. Yeah. So does allotransplantation needs to be done by closely related donors only? Yes. As I told you that your HLA typing is must when you go for allotransplantation. Next question is, cell therapy, we can transplant healthy cells in patient. But what about cells present in the patient? Will they not cause disease again? Yes, very good questions. So it's not like that you are just transplanting healthy cells once. You have to, give therapy to multiple times so that slowly all the disease cells will be replaced by the healthy corrected cells. It's not like that you just give cell therapy to one time. You have to keep on doing several times. And then there is a question, let me see, how to decide the drug dose for screening. So you have to decide, like you have to decide a minimum concentration of a drug or a maximum concentration of a drug, right? Every drug, you know that what should be the minimum concentration and what's the maximum concentration. So that you have to decide first by hit and trial, and by looking at the preliminary results, you can rationale again the dosing of the drugs for your screening. Okay, next is, let me see one more. Yeah, what if iPSCs from disease patient differentiate into healthy cells and disease state is just removed naturally? Well, it's a very interesting question, but generally it doesn't happen because your disease patient, they will have those mutation in your iPSC cells also because they are derived from the disease fibroblast. So they will carry those mutations. So you have to correct those mutations by genome editing in order to make them healthy cells, right? Okay, then what is the accuracy of differentiation of iPSCs using various growth factors? So as I told you that the differentiation or the reprogramming efficiency is very low. So what you have to do is you have to differentiate cells and then you have a stringent criteria to select those differentiated cells. So accuracy is less, but you have a stringent criteria to select your differentiated or your iPSC cells. Like once you, in case of iPSC generation, the efficiency is 0.1%. But once you have those 0.1% cells, you can expand them in trillions and billions. So you should have a stringent criteria for selections. So due to time constraint, I can take only these questions, but as we told you that we will provide you answer of all of the rest of the questions in our separate question answer document. I hope you enjoyed today's sessions, and I request all of you, please join our next session also, which is where we will discuss the applications of these stem cells and induced pluripotent stem cells in healthcare, right? So with this, I can stop myself. Over to Shubham if you want to say something. Shubham, you there?
I'm here. Yeah, so thank you so much for a wonderful session. And so as Ankur suggested, we will answer all the questions later in the form of a document along with the PPT handout. So thank you so much for joining this session, and request all of you that please join the next session also. It will be equally knowledgeable session, and it will clear your concept in the terms of application part in the regenerative medicine part. So thank you so much for attending the session, and thank you so much, Dr. Ankur, for a wonderful session. I hope their doubts and concept on iPSC are very much clear now. Thank you, everyone. Thank you so much.
And I request all the attendees that please join our LinkedIn group because we are updating all our events over there. So if you want to know what workshop, what kind of certificate courses we are running or our future workshops are there, please join our LinkedIn group. The link is already shared in the chat box. If you see initial message, you will find a link over there. Please join our LinkedIn group and get updated regarding our all events and workshop and trainings. Thank you so much.
Thank you, everyone.